Hello everyone, and thanks for joining us today for this webinar on proliferation analysis in FCS Express 7. The agenda of today is pretty straightforward. We will start with a bit of background on proliferation analysis, then we will create an analysis using real data, and we will see how to use controls to improve the analysis. For example, how to set the background level and how to set the starting generation using your controls. We will also see how to retrieve statistics from proliferation plots, and finally how to export your analysis using batch processing.
Okay, so the overall idea at the basis of using fluorescence tracking dye to study cell proliferation is that with each division, the dye is divided roughly equally between the two daughter cells. And so at the end, the dye fluorescence intensity decrease that you see here, so the dilution of the dye, it's basically a measure of the number of division any given cell has undergone. So you see here, you stain the cells, and then you have half of the intensity, half of the intensity, half of the intensity. Again, if you want to speak about a theoretical model, what you should end up with is this one. You see, you have all the cells stained undivided. Then at half of the fluorescence intensity, you have the first generation. And in this theoretical model, all the cells in this generation are all stuck together in the same channel because they have the same amount of fluorescence of dye, okay? Unfortunately, as you may already imagine, the real life it's different. So you have variability, as we mentioned before, because of the instrument, because of the procedure, of the protocol. So usually end up with something like this. So in which each generation actually overlaps to the neighbor generation. And you may also end up with something like this if you mix population together. So let's say that you have like a stained and proliferating population, and then you mix this population with an unstained population. So at some point, the stained cells will proliferate so much that the dye will be so diluted that basically the fluorescence of the stained cells will match the autofluorescence. And so in that case, you will have to find a trick to say the model, okay, do not fit any population which is below this threshold where this threshold is defined by your background, by your autofluorescence. Okay, so let's start. And today I'm going to use this 4 sample. You may have like 400, it doesn't matter. You probably already know that FCS Express, it's really good in analyzing multiple sample iteratively. And you see, I want to briefly describe what those samples are for. So you see an unstained control, which is basically the autofluorescence. Then I have a stained control, but not stimulated. So those are cells that will give me an idea of where the undivided peak, so when the starting generation will be on my histogram. And then I have a control to test that my cells can actually proliferate. So it's stained cells stimulated with a drug known to induce proliferation. And last but not least, I have my sample. So the sample treated with the drug that I want to evaluate. We can just start with any of those samples. Let's say we'll start with the positive control. I will just drag and drop. I will create a density plot. You see we have few parameters here. We also have a staining for CD4. So just consider that you may have any kind of complex mix of antibody. It doesn't matter. What I'm going to select is basically just those two. So you see this is live and dead staining. This is my cell trace. So this is the tracking dye. Let's first remove, for example, dead cells. So in this case, dead cells are positive for this dye. So I will just exclude positive cells. Let's call this live. And then I will drag and drop as we did before. And in this case, I want just to gate on my cells. Okay. And those are my cells. I want to have a quick look to see if my gates are okay for all my sample, and it is very easy. You see I can just scroll through my sample using this double arrow. For example, we may want to do this, for example. We may want to adjust a little bit of this. And I just want to mention, it's not the case, but you see we have a data specific gate button. So if you want, you can tweak gate sample by sample. In this case, I just want to use gates, the same gate for all my sample. So this is pretty easy. FCS Express basically automatically apply this change to all my sample. When you are happy with this, that's it. Otherwise, as I mentioned before, you can just go here, click on data specific gate and tweak this only for this sample. Okay, so this is my gating strategy. Again, it can be as complex as you want. You may have like tons of gates, nested gates, children gates, or whatever you want. What I'm going to do is just opening the proliferation fitting plot. Exactly as we did before for the multi-cycle DNA plots, we have this proliferation plot. And this is the proliferation plot. Oops. Yes. I want to select the cell trace, which is my tracking dice. You see, it looks pretty weird, and this is because we didn't apply a gate yet. So basically, this is ungated. I want to apply this gate, and you see now this is actually my sample. You see, exactly as we saw before, the fitting model actually brings a lot of different overlays. You will see that you can format those overlays. All the single generation here are depicted as a green overlay. The sum of those green population is the red profile. The difference between the red profile, which is the fitted model, and the black profile is the noise. And the noise is depicted by this light blue overlay. I will not go into any detail, but again, if you right click and go to format, or if you go here to format, you can format any tiny detail of this plot. I just want to mention the overlay category. You see each overlay can be formatted just to show you, if you want, all the generation will be filled with green and will be transparent. So this is just an example. Otherwise, you can just undo in this case and go back to what you have done before. I just want to mention that you can do this type of things. Okay, this is the auto-fitting. So the model, what the model is doing is starting from the right, looking at the first pick available. In this case is this one, and this is defined as the starting generation, so the undivided pick. And then based on the theoretical model, it's looking for picks at half of the intensity moving to the left. Usually you cannot fit more than 10. In this case, we have probably 2, 3, 4, 5, 6, 7, 8, 9, 10. Probably usually because otherwise the last pick start to merge with the autofluorescence. So if you want to retrieve statistic from this plot, you can just right click and go here, statistic. You see, we have the histogram statistic, which is a standard type of statistic, like for example, if you create markers. And then we have two specific proliferation statistics. So the fit one, and let's put it here. And last but not least, the population statistic. Let's see if we have a little bit of room below. So the fit statistic gives you summary statistic for what you are displaying in this plot. So you see division index, proliferation index, percentage of divided cells, so percentage of original cells that undergone divisions. The peak ratio, again, ideally speaking, the peak ratio should be 0.5. In this case, it's quite close. Number of fitted cells, number of original cells calculated. So this means that the original cells give rise to this has been calculated to be 4,200. This is the position of the starting generation, which is this peak. You see here, 2,000,007, which is here. And this is the position of the background. In this case, it's not even active. If you want, you can actually display those two markers on your histogram. And you see here, when you if you want to format your plots, we just saw the overlays, but you see here you have, for example, position markers. So you see those two markers, undivided and background, are basically those two. So you can make them actually visible. For example, let's say the undivided peak will be green, will be a little bit bigger, and it's here. And the background, let's make it bigger again as well, and orange. And you see it's here because it's basically turned off. I will show you how you can turn it on. Now, again, we are using the auto fit. It means that if you move through your sample, you see the starting generation, it's changing automatically in order to try to find the best fit. And of course, all the statistic update automatically based on that change. The generation statistic, so this table, gives you a lot of different statistics that you can also increase by right-clicking, like number of original cells in each population, percentage, and gives those population on a generation basis. So for example, generation 1 and 2, you see they are basically 0, and so are here in this part. Then you have generation 3 that contains, for example, 359 events, which is this peak. And those 359 events has been generated from 44, let's say 45 events. Basically, 8.22% of the original cells generate this third generation, and so on. So those are all the single generation statistic. And again, if you scroll through your sample, this is, for example, the undivided. You see it's just one peak. Basically, it's all undivided. But those statistics update immediately in real time. So I want to show you a couple of different tricks that you may use to analyze your cell, your proliferation analysis. So for example, what happens if your cells, if all the cells that you plate actually proliferate? So basically you will end up with no undivided peak. And if this happens, the proliferation fitting model will use as undivided peak the first peak that it will find by searching from the right to the left. So in that case, you may want to fix the starting generation. Let's say that you have like 20 sample, 100 sample. If they all comes from the same experiment, you may have like a control, so the undivided control. And remember, we do have the undivided control. And you want to make sure that all your data, all your sample are analyzed using that control. So this is what I'm going to do. So you see here we have the stained but unstimulated control. So I will open this here. And actually I want to open it as an histogram. Now the software doesn't know which parameter I want to show. So I want to show the cell trace. And let me also apply the gate. And you see here, we have created a cells gate, which is this one. You can drag and drop, or I just want to show you another alternative. You see here, cells gate, this drag drop down menu. So this is the unstimulated but stained control. And what I want to do is to calculate, for example, the median and set my starting generation to that median. And if we want to be even more, let's say, precise, I will do the same for the background. So I will open a new plot. I just duplicate. I will drag and drop my unstained controls. So this is basically showing me the autofluorescence. I'm going to replace the existing overlay. Of course, now this marker doesn't make much sense, so I will adjust it. So this will be the background, and this will be the plots that I will use to calculate the background. So I will take the median for this from this population, and this is the sample, the plot that I will use to calculate the starting generation. So now I know that I want to use these two statistics. How can I tell my fictive model to use those statistics? Well, if you go here to the Format tab, among the many, let's say, aesthetical options that you have, like overlay, axis, title, position marker, you see here we have proliferation fit. And I want to show you the content of this dialog. So this dialog allows you to add constraint or remove constraints to the model. You see here, for example, maximum number of picks. So the maximum is 10 because, again, otherwise your cells basically will eat the autofluorescence, will match the autofluorescence. If you change this, you see that cells, that cycles are disappearing. Sorry, generations. In this case, 10 is fine. Then you can constrain the CV. So for example, you have multiple sample and you want to fix the CV among all of them, or you also want to fix the CV among all picks. This is usually by default checked. You can do the same for the pick ratio. So again, the pick ratio ideally should be 0.5. In this case, it's 0.51, which is fine. You can fix it among all sample. You can fix it among all picks. That's up to you. And you see the last two options here. So starting generation and background noise. So now the starting generation, it's not fixed. It means that it's automatically calculate the software. And this is why, while we scroll through our sample, this starting generation flipped around. And the background noise is basically unused at this stage. So what I want to do is to tell my model to use a fixed starting generation, but I don't want to type a number. I want to precisely base my starting generation on the median of the control here. So this is what I'm going to do. Sorry, let me go back here. I will remove this. I will click on T. T means token. So I'm going to retrieve a token, which is in this case a statistic, from this plot. And the statistic will be the median of the M1 marker. So first of all, which is my plot? It's this one. And the statistic will be the M1 median. Now you see in this field, you don't see a number, you see a tokens, which is gray highlighted. And I will do the same here for the background. I will remove this. I will click on token. Yes, I do want a statistic. And in this case, this is the plot, the unstained control. And the statistic is the median of the M1 marker. Now you see that the marker for the background is set here. And should you change the control? For example, if you want to use this template next week, you will have new sample and you will have new controls. You will be able to use this template again. You will not need to create multiple plots, multiple gates, multiple markers. You will just load your file into the data list, load the file into the right plot, and that's it. So let me close this one. This is a nice way to put a constraint regarding the autofluorescence. This means that should you face a sample in which you have a negative population, this is a way to tell the software, please stop to fit my proliferating cells once you reach that value. So this is our analysis. What I want to do now is show you how you can, for example, batch this analysis through different sample. So basically, you see here we have multiple sample. I want to fix these two overlay in order to have them, to keep them always fixed while batching through my sample. So I'm going to select them, click on overlay. You see here by default on all plots we have this option, change data file during next, previous, and batch. And by default this option is checked. This means that every time that you load a new sample or every time that you batch, that overlay will move to the next one, to the next sample, or to the previous sample. In this case, I don't want to do that, so I will uncheck this. So here you see I have my data list. This means that now, if I switch through my sample, only the plots in my page will change, while the plots, the histogram that I use as a control will not. So you see, this is basically the autofluorescence. We can probably tweak a little bit the model to make this to be pure background. I just want to remember that we have much more options here. We are not going through all of them, but you see here you can put a constraint for the CV. You see the CV in this case is super huge. You can put a constraint for the peak ratio, which in this case is pretty far from the theoretical model. But again, I think you definitely get the point. This is our unstimulated control, and then we have the positive control and our sample. So let's say that we want to export our sample and the stimulated control, and maybe we want to export to Excel and maybe to PowerPoint. The best way here is batch processing. You see here in the batch tab, we have a batch actions. So batch actions are the type of export that you want to perform automatically every time that you run the batch. And those actions will be saved in the layout. So again, if this is your template, next week you will load new data, you will populate your plot, you will probably need to adjust a little bit the gates, the marker, but all the plots, all the gates, all the marker will be there, and also the batch action that you are going to create will be there. So no need to create them more and more times. And again, I want to show you the PowerPoint because I think it's pretty useful for most of you. You see, we're going to fill this dialog, we have few options. For example, I want the presentation to automatically open. I want to export this page. I want to combine all the iteration into the same presentation, so we'll end up with just one PowerPoint. And in this case, I'm going to export only 2 files. If you have 200, doesn't matter. The software will export all those files for you. I'm going to set an output. So PowerPoint batch export, and I want to save it on my desktop. And that's pretty much what you need to do for the PowerPoint. I also want to show you the Excel. You see here we have two types of Excel. The first one, column mode, means basically combine the statistic from multiple iteration together in the same Excel. So this is what I'm going to show you. And at this stage, you see we are start with an empty file. Otherwise, you can also combine if you have an Excel template. And again, I'm going to save it on my desktop. And that's file. You see here we are opening the file after saving. So now we can actually tell FCS Express which are the statistics that we want to export. And we can do this very easily with drag and drop. So for example, let's say that you want the amount of cells within the live population. Okay, you can just drag and drop this gate on top of this Excel column mode, release it, and then you see the cells gate, it's already selected, and you can just select what you want, which statistic you want. For example, percentage, number of events, file name. Those will be 3 columns in my Excel. You can also drag and drop statistics from this type of tables. So for example, if I want all of those, just select them and drag and drop them. I can also drag and drop a whole column. So doesn't matter. Those will all be columns in your Excel spreadsheet. And the amount of row will be will match the amount of iteration that you're going to export. So if you want to export all those iterations, just start from the first one. In this case, I'm just interested, for example, on the last two on my sample. So I will just export number 3 and 4. So we'll start with number 3. I want to show you a few options. For example, here you see in the batch options, we have the ability to pause between iteration. And this is great, for example, if you want to tweak gate and markers and quadrants between different iterations. So basically what the software will do when you will hit batch. So let's do this. Let's start so that we have at least 3, otherwise it's really not enough. We'll stop after each iteration and we'll give you the chance to tweak your gate. When you're ready, you can just click on run. Now the first iteration will be exported and then the software will stop on the second one and will give me the ability, the time to tweak my gate. So if I want to do this, I can do it. I'm just doing something crazy so that you will be able to see the difference in PowerPoint. And we can define whether we want to keep these changes for the next iteration or whether we want to restore. Let's keep these changes and let's continue. So this is iteration 3 now. The software is stopping here. And let's say that we want to do, I don't know, this type of change. Again, I'm just exaggerating so that you will clearly see a difference in the PowerPoint. And then I will continue. And then the software will automatically open my PowerPoint and my Excel. So this is the PowerPoint and this is the Excel. So let's start from the Excel. I have exported 3 iterations. You see here the control, the positive control for proliferation and my sample. Each column has an header. You can actually customize this header if you want into FCS Express. Otherwise, you can just keep the default one. You see we have all the statistics that we have exported are here presented as different columns. And all the statistics make sense. So for example, this is, you see here, it's the undivided peak. And in this sample, which is the unstimulated, it's 100%. And all the other generation in this sample are 0. And then you have the positive control, and then you have the sample. And in this case, 2 population are 0, and then you have all the cells, and so on, and so on, and so on. And as I mentioned before, if you have a template like this and next week you want to export right down here, you can do that without starting with a new Excel. Let's have a look at the PowerPoint also. You see we decided to combine multiple iterations into the same PowerPoint. So this is why we have 3 pages, 3 iterations, 3 slides. This is iteration 1. Then we stop, if you remember, on iteration 2 and we make some change. You see in this gate. And then we stop on iteration 3. We keep those changes in this file and we add different changes on this plot on the sales gate. I want to mention that all those plots looks really great. You see they are super high resolution. This is all vector graphic. I also want to mention that those are ungrouped. You see, you can move them around. You can also define whether you want to keep the plot that are out of the page or just hide them. If you are on Windows, you can also ungroup them. And you see basically now everything is ungrouped. I usually suggest if you want to tweak something like change the axis title, change the aesthetics, it's much better to do it in FCS Express, because if you need to spend time here and you have 10 files, you need to do it on 10 plots. While you already know that just select one plot, and here in the formatting option you have a lot of different aesthetical options that you can use to make your plot much nicer. Um, Again, I think this is pretty much what I wanted to show you. Um, I want to just summarize. Oh, I want to add something actually, which is pretty important. When you format your proliferation plot, we already seen, for example, that you can format the color and I don't know, the feel of each population. We do this before. I want to mention that, again, you should not smooth your histogram. You may be tempted to smooth histogram because they may look nicer. But again, as we mentioned in the DNA cell cycle webinars, it's definitely better if you let the software fit the raw data, not the smooth data. So if you want to make your a picture nicer, just do it on another plot and retrieve the statistic for the fitting calculated on the unsmooth data, which is much better. Okay, I want to conclude just by saying, let me zoom out a little bit. It's super huge. I want to conclude saying that proliferation fit model are really easy to handle in FCS Express. It's basically just a plot type among many other plot type. So you can insert as a standard plot type, you can format it. So if you know how to use a standard plot type, you will also know how to use a proliferation plot. You can format that plot in many specific details. Remember, number of fitted generation. You can put constraints on the CV, on the peak ratio. You can set your undivided cell peak, your background threshold. So this is really a powerful implementation of proliferation fitting model. And then you can integrate this with any other tools we have. So for example, batch processing, as we did a few seconds ago. So I think it makes really the difference if you're dealing with proliferation analysis. And again, I want to mention that on our website, you can find a lot of different information. I want to just mention this chapter of the manual. You see here in the manual, we have a proliferation plot chapter and we have a proliferation statistics chapter. And I want to show you that all the statistics calculated are depicted here with all the mathematical formula, like you see here: proliferation index, division index, percentage of divided. So we are pretty transparent on the way we are calculating those statistics. And I usually suggest you to have a look at this because there is not really consensus in the literature on what the proliferation index is. So if the reviewer asks you, how did you calculate it? You can just go here and share with the reviewer this formula, and just to be super transparent. Okay, I think this is what I want to show you. Again, if you want to try FCS Express and the proliferation model, just download the 30 days trial. It's fully functional, and you can play with your data as much as you want.